Hi everyone. Let's just wait for maybe 2-3 more minutes and then we'll start. Okay, so I think we can get started. I'll just share my screen. So I think my screen is visible to everyone. So, right. So I'll get started with the problem solving session and covering some interesting topics that were uh, sort of brought up in the coursework of week 7. So week 7, as you guys must know, it covered topics from aging, senescence, cell death, and then there were 2 special lectures on apoptosis, which went into details about the 2 different pathways of apoptosis, intrinsic and extrinsic. And you also learned the difference between apoptosis and necrosis. And of course, the hallmarks of aging and the 7 to now 9 pillars of aging and various characteristics that senescent cells would demonstrate, would show in culture. And yeah, and also, of course, about the cancer cell and what are the hallmarks of cancer. What would you see in a cancer cell that's different from a normal cell or an untransformed cell? What is the tumor microenvironment? What are some factors that are present in the tumor microenvironment that make it so inhospitable for immune cells? What are cancer stem cells? And what are some ways in which the cell cycle is regulated in cancer and how it's differentially regulated with respect to some mutations, some gain or loss of function mutations in cancer, right? So that was I'd say, a interesting week of lessons, and I hope you guys found it interesting. I'll cover some topics related to that. So as you all saw in your coursework, um, there was a definition with respect to what aging is, what senescence is, what is cell death, what's programmed cell death, what is inflammatory cell death, right? So we know that aging is a time-dependent accumulation of cellular damage. So over time, cells are dividing, but there can be genomic instability, as you can see here. Genomic instability, telomere attrition, epigenetic changes, loss of the proteostasis, macroautophagy can be disabled. There can be deregulated nutrient sensing from the environment, mitochondrial dysfunction, cell senescence, of course, stem cell exhaustion, so you don't renew new cells, altered intercellular communication, chronic inflammation, which I'll go into in a little bit in a couple of slides, and dysbiosis. So I'll just explain these. Of course, you guys already have an idea of all of this, I'm sure. So genomic instability, as we know, that as people age, there is an accumulation of sometimes mutations or damaged DNA, and where the response of the cell to repair that cell or repair that break in the DNA or that damage in the DNA or send the cell to apoptosis becomes less efficient. So this can lead to genomic instability over time, and a part of that can be because of telomere attrition. So telomeres are the ends of the chromosomes, which are tough to replicate, which is why typically you can add some extra base pairs there that allows it to replicate. But what happens over time is that these telomeres actually start getting smaller. And what would happen if the telomere starts getting smaller, like towards the end? This is like the telomeric sequences are protective sequences, but if they're getting smaller, what would happen? These ends would start getting exposed, and these regions can be where you can have some you can have joining of these ends. You can, and they're exposed to various different uh, threats with respect to genomic stability. So telomere attrition, epigenetic alterations. One example of this is change in methylation pattern of the genome, which I'll go into in a little bit. And also, of course, it can affect histones and their functioning when it comes to um, sort of the transcriptional balance in the cell, what cells, what genes are properly, like the gene expression regulation can be changed with respect to histones functioning correctly in packaging the DNA or allowing certain regions to be uh, more like euchromatin, some regions to be more like heterochromatin. Loss of proteostasis is pretty self-explanatory. Essentially, you just don't have um, the the correct response to, say, unfolded proteins. So if you have some badly folded or misfolded proteins, or some product of, say, damaged DNA, ideally the cell should respond to that as well and send it off for degradation so it doesn't harm the cell. But in aging cells, what may happen is that this unfolded protein response and other sort of damage response pathways are altered and they're not as efficient anymore. Macroautophagy can be disabled in the case if there's a damaged organelle. Getting rid of that to protect the cell as a whole is a very important process, but that may be halted. It may not be as efficient. Nutrient sensing makes sense, right? That you cannot sense the nutrients in the environment or in the tissue fluid, and that makes the cells less metabolically productive, leading to a lot of different problems. Mitochondria can become dysfunctional. They're not um, uh, efficiently performing all of their many functions anymore, and that can encompass like a whole host of different fusion processes can be messed up, or you can have mitochondrial replication being messed up. The list is endless. And then cell senescence, of course, we'll go into that. Stem cell exhaustion, so your pool of fresh cells, they are no longer being as frequently renewed. So the old cells are all you have, and if they die, then there's a much slower sort of replenishment of those cells. Altered intercellular communication. This can be, for instance, in the immune system, it could be that suddenly cells are more reactive and there's chronic inflammation, which is said here. And dysbiosis is with respect to the gut. So you can have, as you guys know, the gut microbiota and other microbiota, various different organs in the body are actually very important. And having symbiotic or commensal bacteria in these organs can really influence many different important functions in the body. Like they can influence metabolism, it can influence gut health, um, and there's like a gut brain axis that can influence, you know, your hormonal secretions, endocrine system, so on and so forth, moods, etc. So there's, so if there is some imbalance in the gut, if there is some change in the composition of healthy bacteria in the gut, that can lead to dysbiosis, and that can lead to a whole host of different problems. So all of these, unfortunately, when people are aging, these can start affecting people in different ways. So this is, yeah, this is just like a summary of all of that that was also explained in your coursework. So one of these things, like I mentioned here, was epigenetic alteration. So I thought I would go into it in a little more detail because it's useful to understand. So as we know, in the genome, in the mammalian genome in this case, I'm saying, the DNA methylation is an epigenetic mechanism that is very important, and it actually involves the transfer of a methyl group, so a CH3, right, onto the C5 position of the cytosine to form a 5-methyl cytosine. Okay, so this DNA methylation can actually regulate gene expression. It can either recruit proteins involved in gene repression, or it can inhibit the binding of transcription factors to the DNA. So if you have a methylation over here, it can actually reduce the transcription of this particular stretch of DNA. Although, of course, it is a little context dependent, sometimes it can enhance also. There are cases of that that also have been studied. Yeah. So during development, you have a certain pattern of DNA methylation in the genome, and with time, it's like over time it changes. And this can be because of de novo DNA methylation and demethylation as well. So over time, what do we need basically as an organism is developing, as it is, as the body is changing, as the body is aging, you're going to have, different requirements in the body, right? So certain genes will no longer be required as much, or the products of those genes will not be required as much. Some will be required more, or some will be required in different regions. It could be, right, the way bones lengthen, or the way eventually some bones stop growing completely in comparison to when people are kids, right? So you have, so behind all of this, there's a lot of activating and deactivating of DNA, and one of the processes involved in that is this pattern of DNA methylation in the genome. So with time, since it's dynamic and we're methylating and demethylating DNA, differentiated cells develop a stable and unique DNA methylation pattern that regulates tissue-specific gene transcription. So essentially, if you have a differentiated cell that, say, like an osteoclast, okay, that's like a bone cell, it will have a certain stable and unique DNA methylation pattern. So it's the genome in that particular cell in the bone will have a certain pattern of DNA methylation, and that will affect the tissue specific gene transfer. So in the bone, some specific set of genes will be, so you don't need every single gene to be transcribed actively in the bone, right? You just need the ones that are relevant in that tissue. So that's how the gene transcription will take place. And the process is easy. It's just DNA methylation, which is catalyzed by a family of DNA methyltransferases. So what they do is they transfer this methyl group, CH3 group, from SAM, S-adenyl methionine, to the fifth carbon of cytosine residue to form this 5-MC, okay, 5-methyl cytosine. So here this process is shown. So there are two ways it can happen. One is these de novo DNA methyl transferases that transfer methyl groups onto naked DNA. So you just have a DNA stretch and they've transferred it. So you have DNMT 3A and 3B that do this. And then there's another one called maintenance DNMT. This DNMT1 here is involved. So what it does is, suppose you have DNA that is replicating semi-conservatively, right? So one strand, the parent strand is going to have that particular methylation pattern. Now, if you were to create a whole new strand, that's not going to have the methylation pattern. So this DNMT1 comes to where this replication fork is, and you're getting this new DNA strand, and it starts adding methyl groups at the relevant positions in that growing daughter strand so that it can replicate the methylation pattern as well. So this is two ways in which DNA methylation occurs, right? So I hope that is clear. So what is the relevance of this DNA methylation, right? So as I mentioned, of course, with aging, it can actually affect the gene expression patterns and the level of different genes that are expressed, right? So this is epigenetic information here that we're talking about. So during aging, this epigenetic information changes sporadically in response to both exogenous and endogenous factors. So from factors both from outside and within the body, okay? The resulting abnormal chromatin state is characterized by different histone variants being incorporated, and they can have altered DNA methylation patterns and altered histone modification patterns, which result in the recruitment of different chromatin modifiers. So essentially what this is saying is that you can have not only altered methylation pattern, it can also change with respect to the histone variants that are incorporated while packaging the DNA, and different histone modifications, like we had seen last time, that histones can be acetylated, deacetylated by HATs, HDACs, and so on. So the histones themselves can be modified differently. DNA methylation patterns can change. Different histones themselves or their variants can be incorporated. And all of this will result in different ways in which the gene expression can be changed. And in older cells, you can actually have abnormal chromatin state, which can include these altered transcriptional patterns or transcriptional drift within the population, and that can lead to genomic instability and mutations. You can have these transposable elements that are inserted in the genome, and yeah, and cause some mutations to occur in the genome and cause genomic instability, right? So that's one way in which the epigenetic changes during aging can cause these various instabilities, leading to a transcriptional drift and an altered state of the cell altogether. So I hope that is clear. So this is an interesting point that I thought I would bring up. So while you guys were reading about aging, right, we know that, aging has some hallmarks, as I mentioned, right? That the pillars of aging that I went across. So one of them was that you can have some dysregulation of nutrient sensing. So your metabolic pathways can go haywire, or certain cells can start performing really weirdly, and that can actually change the state of the tissue. It can lead to tissue degeneration and so on. So there was an interesting so of course you guys read about how when it comes to nutrient sensing pathways during aging, or in general also, we have four different pathways that are the major ones. You have some anabolic pathways and some catabolic pathways. So anabolic are the ones that respond to um, that essentially create, right? They build, and if you have and they are associated with aging. So you have too much of this upregulation of anabolic pathways, you actually have aging. Whereas other pathways, so that anabolic pathway includes mTOR, and then you can have AMPK and sirtuins that are highly implicated in catabolic processes, and they respond to, and they are related with healthy aging. So there's interesting research now, and there have been some clinical trials as well done on both humans as well as primates, non-human primates like monkeys. And in them, they've seen that calorie restriction, which is a process in which you reduce your dietary intake below the energy requirements, but you maintain optimal nutrition. So you don't lose out on the important components of your meal. Like you should have carbohydrates, fats, proteins, vitamins, minerals, all of that should be balanced. It should just be less than your daily energy requirement. And this apparently is a type of nutritional intervention, like a type of diet essentially that you can follow, which has the potential to attenuate aging. Of course, I have to put a disclaimer there that this is just some preliminary research, and while it is, it has shown some positive results, one has to remember that every person is different and everyone's composition is different. So what works for one person may not work for another person. And especially when it comes to people in specific, like what do you say, like special circumstances, or like more complicated circumstances, like with somebody with comorbidity or someone who's old, very young. Of course, it would be different in that case. So what people have seen till now, anyhow, is that calorie restriction can reduce mTOR activity. So just now I mentioned that mTOR is an anabolic pathway, so that can lead to, that has seen that if you have high mTOR activity, that can lead to some aging-like characteristics. And so if this CR or calorie restriction were to reduce mTOR activity, you could have decreased protein synthesis, which reduces cell stress. And cell stress, we know, like ROS production and so on, can lead to senescence and aging-like phenotypes. And here also they saw that there's enhanced autophagy, and that is, as I mentioned, a process where your damaged cell components and organelles are recycled. So this can actually improve cell function. If you're removing damaged parts, you're going and recycling them to produce new and more functional parts, you'll definitely improve cell function. So you want to have more autophagy, like efficient autophagy, within the limits of reason, and you want to reduce cell stress. So CR reduces mTOR activity. CR also activates AMPK, as I mentioned, that is the catabolic pathway. It can inhibit mTOR, of course, and it can also have a profound effect on mitochondrial biogenesis and improve your energy efficiency. And I think I forgot to add another word here, but it can reduce ROS production. Yeah. And of course, you guys have heard about sirtuins, right? People try to boost sirtuins by eating superfoods and saying sirtuin boosting foods like chocolate and grapes and wine and whatever. So there are the sirtuins are of great interest in the aging field. So calorie restriction has been shown to increase NAD plus levels, which actually activates SIRT1 because it's an NAD plus dependent enzyme. I think it's a DS-stylase or something. And that can regulate, of course, metabolism, stress resistance, and DNA repair. So maybe, when people are aging, if they want to sort of look at the metabolic, sort of molecular side of things, this is one thing that they could try, which is calorie restriction. Of course, under supervision. So it's just an interesting sort of clinical aspect of molecular research. Right. So aside from these points till now, in the, where is it? Yeah. In the hallmarks of aging, we also have chronic inflammation and altered intercellular communication. And these can actually include a lot of issues that develop in the immune system of a person. So I'm going to talk about two things. One is inflammaging and one is immunosenescence. So I'll explain what they are and why they're relevant here. So inflammaging, it's basically inflammation and aging as a word, it's just being squashed together. So during aging, chronic, sterile, low-grade inflammation occurs in the body, and this is called inflammaging. Okay, and this develops um, over time as a person ages and contributes to pathogenesis of a lot of age-related diseases. Okay. So some potential mechanisms of inflammaging include genetic susceptibility, central obesity, increased gut permeability, changes to microbiota composition, like I mentioned, dysbiosis, cellular senescence, then you have inflammasome activation, oxidative stress because you have dysfunctional mitochondria, immune cell dysregulation, and chronic infections. So all of that is sort of encompassed over here. So essentially, it is an immune dysfunction because you constantly have some amount of inflammation in the body. So there may not be any, pathogenic agent. They're saying it's sterile inflammation. So there is no pathogen, there's no virus, no bacteria. It's just the cells, immune cells reacting constantly in a dysfunctional way. They should not be reacting unnecessarily like that. And that leads to an accumulation of tissue damage. It can lead to um, a, a loss of healthy cells. It can lead to DNA damage. It can lead to even cancer because if you have a constant chronic low-grade inflammation, actually it can erode the immune system enough that cancer might develop. So inflammaging is another up and coming sort of topic of interest in aging research. Uh, Hand in hand with inflammaging is immunosenescence. So as we know, with time, with aging, it's a bit weird to think that if someone has chronic low grade inflammation, why is their immune system actually senescing, right? Why would their overall functionality of the immune system go down when they have continuously got inflammation? That's because the good functions of the immune system actually start eroding, and you have therefore bad consequences. So for instance, you can have impaired immune responses of your various different immune cells, T cells, NK cells, B cells, macrophages, myeloid derived suppressor cells, and so on. So you can um, have, say, not as effective an immune response to a threat as required, or you can have repressive effects on immune cell activation and proliferation, leading to an impaired immune response. So the body is not able to fight off pathogens as well while the person is aging. And this will result in these different diseases, neurodegenerative diseases, autoimmune diseases, where suddenly the body starts fighting itself, or it can lead to tumorigenesis because the immune system, essentially, when there is some DNA damage or some accumulated mutation that causes the cell to start dividing uncontrollably, suddenly the immune system, because it has not got an optimal immune response, it cannot detect this tumor. So it starts growing and growing. And that is a hallmark of immunosenescence. And this can be because of thymus degeneration. So thymus is an organ where T cells mature. So if you have the thymus degenerating, you will have a lot less of these naive T cells that will come out of the thymus. You can have, as I mentioned earlier, inflammaging that includes this SASP, or senescence associated secretory phenotype, and cell senescence. And then you have this low grade amount of these various inflammatory cytokines. And then your population of immune cells, here they've given the example of T cell population. And as I mentioned, because the thymus is degenerating, you can actually reduce the amount of naive T cells and effector T cells, which are the ones that actually go and kill infected cells or cancerous cells. Your diversity goes down, so the ability to react to different kinds of antigens goes down, and you just have a bunch of memory cells increasing in number. But what is the point of memory if something new comes into the body, right? You can fight what was already there, but even those cells actually are not always the most effective. And if they cannot differentiate into effector cells or effector memory cells, then they won't be able to kill off the threat anyway, even if they recognized it. And of course, you can have these many metabolic changes, which is a sort of overarching problem during aging, which is uh, metabolic dysfunction, increase in ROS, and decrease in mitochondrial biogenesis. So this all can lead to immunosenescence, which can then cause a whole host of diseases and disorders uh, in an age-related way. Yeah. Cool. So another point I thought would be interesting for you guys to visualize is the mention about DNA damage foci, right? So we in your coursework it was basically it went into detail about the characteristics of cells that are senescent or aging or have undergone some sort of damage or got some sort of damaging stimulus like that. So, of course, one is that cells will be in cell cycle arrest. They will not be dividing. They'll be in cell cycle arrest irreversibly. The DNA damage response will go up, and you can have various different phenotypes that show up, such as telomere dysfunction-induced foci, these TIFs, or DNA scars, where you have some chromatin alterations in some segments. You can have the SASP, which can be assessed by various different proteins that are secreted or on the surface, epigenetic changes, and one particular one out of all of these different characteristics are these DNA damage foci, which have these enriched gamma H2AX amount. So what are they? So the DNA damage foci are discrete nuclear regions where DNA repair proteins accumulate in response to DNA damage, particularly DSBs or double-stranded breaks. So at these foci, they're going to be like a signaling hub that recruit DNA repair machinery to that damaged site. And they can be visualized with some fluorescence microscopy. So you can stain them with antibodies, anti-gamma H2AX antibody, for instance, can stain one of the DNA repair machinery related proteins at the damaged site and can help you look at them, and they look like punctate foci. So here you can see these are nuclei stained with DAPI. I believe these are E549 cells, so these are lung carcinoma cells. And then you have these gamma H2AX foci. This is the control, and then they started increasing the concentration of this drug called etoposide. Etoposide, I think, is I think it's a topoisomerase inhibitor of some sort, and when you use it on cells, it causes a lot of double-stranded breaks. So as they started increasing the concentration of etoposide, you can see that these foci are becoming more and more prominent. And you can see the punctate gamma H2AX staining. So what is gamma H2AX? It is a phosphorylated H2AX. So H2AX is a variant of histone 2A, and it's very important in the cell when it responds to double-stranded breaks. So when there is a DNA break, this ATM, remember I had talked about the ATM protein and how it's important in double-stranded break repair. So this rapidly phosphorylates serine 139 on H2AX. Okay, and this will give you this gamma H2AX. Essentially, it is a phosphorylated form. And this event spreads across various different stretches of DNA at the break site, and it creates this platform for DNA repair protein recruitment. So then it can call all these other repair proteins like BRCA1, MDC1, and so on and so forth. So you can even stain with these based on what tissue it is, what type of cell it is, you will have different machinery. But this gamma H2AX is a well-known marker for the DNA damage foci, and that's what it looks like. Okay. So yeah, that I thought might be interesting for you guys to see. Okay, so we're starting with the questions now. So first set of questions, do you guys have anything you want to ask me? Any questions, anything confusing you? You can let me know. Otherwise, I'll move on to the questions. No? That's good. Then I guess I can move on with the questions. So if anyone is joining this problem solving session for the first time, I'll just let you know that when we go through the questions, I read out the question and then you guys can either unmute and say the answer, or you can send the answer in the discussion chat box thingy. So that's up to you, whatever you're comfortable with, and then we'll discuss what the answer is. Okay. Right. So here is one question to start off. What is the primary characteristic of aging? You guys know? Anyone? This is an easy one. Yep, I've got one answer. Is there anyone else who wants to try? Okay, so all right. I think I've got two answers here, and you guys are right. It is B, which is progressive decline of an organism. So that's very good. So why are the others wrong? Obviously, unlimited cell division, while it might happen in a cancerous condition, that's not a characteristic of aging. Enhanced immune response, again, is not necessarily something that someone sees. In fact, it's probably the opposite. And increased metabolic activity is definitely not a sensible readout here. Okay, next question. How does senescence differ from quiescence? Let me know. Okay, I'm getting some answers here in the comments. Anyone else wants to try? Yeah, so you guys are right. Absolutely. Yep. All three of you are right. It is indeed C option, which is that senescence is irreversible while quiescence is a reversible state. So, why is that? When a cell senesces or when a cell becomes senescent, it is actually terminally differentiating. It is no longer going to divide, and this is not a reversible state. Although, of course, I mean, if you look into, recent research, people are trying to do things like, reversing senescence and all that, but I'm not sure myself how successful everyone has been till now in that. And quiescence, as you guys know, is like, the G naught stage, right? The G0 stage, as a part of interfaces somewhere where a cell can go and essentially not divide for some time. It could be because of lack of nutrients, or it could be because of some signaling or crowding or something, and that can lead the cell to make a decision that it's not optimal to divide at that time, so it can go into quiescence. And then later, in some time, if the environment or the conditions were to become optimal again, it can reverse back into interface and start undergoing mitosis again. So, or cell division in general. So that makes it a reversible um, phase in the cell can be in. Yeah. So great. Good. I'm glad you guys all know this. Okay. So now what is the role of telomerase in preventing telomere shortening? What do you guys think? Anyone? Yeah, I'm getting one answer. Yeah. So you guys are absolutely right. It is A option. It adds repetitive nucleotide sequences to the ends of the chromosomes. And this allows duplication of those ends. Why is that? Because we know that at the end of the chromosome, we're going to have a region where you have to essentially make a primer and extend. But if that region is so short, it's difficult to make a primer and extend in that region. which is why after the important gene sequences at the end, we put some repetitive stuff. So that's what telomerase does, right? It puts some non-essential stuff. It doesn't code for anything, these repetitive sequences at the end, and that allows that particular region at the end of the chromosome, that those genes and so on, to be replicated. And it also, it's twofold. It doesn't just help with replication at the end, but it also protects those ends. So from DNA damage and from end-end joining of the chromosomes. And end joining of the chromosomes like that can lead to a lot of different genetic problems. So yeah, so I'm glad you guys know this. All right, let's move to the next question. Which nutrient sensing pathway is associated with promoting aging when upregulated? So I was talking about this earlier. Anyone? Anyone has any idea which one it could be? Hmm. So I've got one answer. Anyone has any other answers that they want to try? No? Okay, so I'll just tell you guys. So actually the answer here is C. So why is that the question? This is, I mentioned this is about promoting agent when upregulated, meaning this pathway is functioning more, and for that reason it is causing earlier senescence, or it's making the cell age more. So AMPK, as I mentioned, is a catabolic pathway. So actually this is not the answer, because if you upregulate AMPK and you have higher catabolism, that has been shown to be associated with protecting from aging. So it's actually good to upregulate AMPK to a certain limit. Sirtuins also, as I mentioned, SIRT1 through 7, these are important for some anti-aging functions. Autophagy is also a healthy process of the cell where it will essentially degrade damaged organelles and damaged parts of the cell, and then recycle all those raw materials and ingredients of those cells to improve the cell. So autophagy, sirtuins and sirtuin activity and AMPK pathway, these are actually anti-aging when it comes to when they're upregulated, okay? On the other hand, this insulin signaling pathway is actually an anabolic pathway, just like the mTOR pathway, including mTOR C1 and 2. These are anabolic pathways. So when these are upregulated, they can actually lead to a lot of ROS production, which can promote aging. There are other processes as well, but one of them is that there is an increased ROS production that can promote aging. So the answer here is C option. Okay. So I hope that is now clear to everybody. I think I have some more questions. Yeah. So what is the key difference between apoptosis and necrosis? Anyone, any idea? Yeah, so I'm getting a lot of answers. Great that you guys are all participating. So you all are right. Oh no, except for the last one. Everyone has said C, so good job. I'll explain why it is C and why it's not the other options, okay? So A option, which is apoptosis, involves cell swelling, while necrosis involves cell shrinkage. This is a very sort of all-encompassing statement, and it's not very true because cell swelling is seen more in necrosis, where the cell will swell and then essentially burst, and a lot of different sort of inflammatory or immunogenic components will fall out of the cell and cause an inflammatory reaction in the body. Okay. So cell shrinkage is more seen in apoptosis, I'd say, because the cell sort of shrinks within itself, produces these smaller apoptotic bodies, right? Necrosis is an active, apoptosis passive, there's nothing like that. So C option is the correct one, which is that apoptosis is programmed cell death, meaning the cell meant to die. Okay, so there is a genetic component. There's a certain set of genes that were essentially transcribed, and the whole process was set into motion as part of a plan to die and not cause too much inflammation. So that's why it is contained, and a lot of these immunogenic proteins that could come out, like excessive DNA, if it were to come out of the cell, that could cause inflammation, right? Or a lot of lipids, if they were to come out of the cell without being covered in a membrane or something, they would lead to inflammation. And if you have a lot of chronic inflammation in the body, again, as I mentioned, it can cause tissue damage. Whereas necrosis causes inflammation, that is correct. So necrosis will occur when it's not a planned or a programmed cell death. It's the cell will swell because of some internal issue. It could be in an infection. It could be the lack of nutrients suddenly leading to this, or some other genetic problem. And that causes inflammation, because if the cell bursts, there can be lipids, proteins, DNA, all of that can come out and attract a lot of immune cells and cause an inflammatory burst in that region. And it is not D, because apoptosis is the one where you have these apoptotic bodies that come out of the cell and that protect the body from excessive inflammation. Yeah. So D is actually the opposite of what is the correct one. So here the option is C. I hope that is now clear to everyone. So what is the difference between these two processes? Okay, here's one question about Hutchinson-Gilford progeria syndrome, or HGPS. Which mutation, like which protein that's mutated, causes this? So let me know if you guys remember. So does anyone remember what happens in HGPS? Anyone know? Yeah, someone has answered, and you're right. It is C option, lamin A. So that's great. So in this disorder, from what I have read, because I'm not that familiar with it myself, there is a mutation in lamin E that can occur. It's pretty rare actually, and I don't think it is like inherited. I think it occurs randomly. Um, and there is a progressive loss of lamin, and so the nuclear envelope is not um, as reinforced because it does not have the correct lamins, and that leads to this premature aging phenotype. So there was an image in your coursework of a person with an HGPS, or I think some related syndrome. And this person is young, but their features are very much like that of someone who is a lot older. And that is because of this unfortunate mutation that causes all of these symptoms to happen a lot faster. Um, and just a lamin A mutation can cause all of that. So it's very interesting to think how the nuclear membrane, if not reinforced well by the proteins within it, can lead to such a drastic phenotypic difference in a person with that mutation. So yeah, so lamin A is the mutated one here. And I think also lamin C might be involved here as well. Though of course, if you guys are interested, do read up on it. If you want to make sure about what other proteins might be involved. Right. So, what is a key feature of senescent cells? What do you guys think? Anyone? Hmm. People are saying B. Hmm. So B is not right, unfortunately, because senescent cells essentially are cells where the telomeres have become very short. Okay. So why would the telomeres become short? Because the enzyme that adds those repeats to the end of the chromosomes, that enzyme is not functioning as well over there. Okay. So you don't have telomerase activity, which is why those repeats are not being added, which is why with every replication you have depletion in the length of the telomere. So actually, if you were to think about it, the key feature or like a feature of senescent cells is that the telomeres are short. So you don't have telomerase activity. And of course, unlimited proliferation is not the case either because they do not divide. Increased mitochondrial function is definitely not the case because typically in senescent cells, they've accumulated damaged and accumulated a bunch of non-functional organelles. So the answer here is C, which is resistance to apoptosis despite DNA damage. So what is that, right? So I think it was mentioned in the apoptosis lectures that there are cells which will accumulate DNA damage in these senescent cells, but they are not necessarily immediately becoming apoptotic, right? So you would think that if a cell accumulates some DNA damage, immediately it would undergo apoptosis so that it's not harmful to the rest of the body. But that is not the case when it comes to senescence. These cells are not going to divide anymore. So even if they have some DNA damage, ideally they shouldn't be a threat. So they have developed ways in which they can protect themselves. For instance, neurons, as was mentioned in your coursework, right? They are not apoptotic, right? They have very low level of these pro-apoptotic proteins and genes that are functional in the cells. And neurons don't divide, and so they won't undergo apoptosis, but they might accumulate DNA damage for some reason. But as long as they're functional, they are resistant to apoptosis. Similarly, you also have cells that are important for structural maintenance. So like you guys know about, the epithelium, right? So you can have cornified epithelium and all that now in your skin. So those cells that are cornified, they are actually dead, and they could accumulate and in the process of them dying and essentially becoming permanently permanently sort of senescent and then eventually dead. In that process, they don't undergo apoptosis and they don't undergo clearance. Why? Because even in that state, they're important for maintaining structure. They're important for maintaining a barrier of the skin barrier to pathogens and stuff. So, senescent cells or terminally differentiated cells that no longer divide, right? They have reached a point where they can be around, but they will not necessarily be um, undergoing apoptosis immediately. Okay. So the answer here is C. Okay. I think last question before the next set of topics. Which of the following is a common effect of DNA methylation on expression, gene expression? Anyone? This is kind of drawing from what I taught you guys about DNA methylation as an epigenetic change and how it can affect gene expression patterns while the organism is developing and aging. So, do you remember uh, what you have either studied before or what I said about?
It's B.
Sorry?
B, ma'am.
B. Hmm. So you're like half right. It definitely does decrease gene expression during aging, and what from what I read in literature, it seems that the overall pattern when it comes to aging is that there is a global decrease in gene expression during aging. However, this is not the only thing. It is not always decrease, right? This is because this statement is that it always decreases. So actually the answer here is C. It can either increase or decrease gene expression depending on the context. Yeah. So, you're part right. So good job. And yeah, everyone who's messaged also, I think most of you guys got it right. That there is both possibilities, but yeah, definitely during aging, overall, there what people have seen till now is that there seems to be a decrease in gene expression. And oh yeah, so I don't know if you guys have heard about this, but it's super interesting. So maybe you can read about it, in your own time. There's something called an epigenetic clock. So essentially, people have seen that with respect to the methylation pattern in someone's genome, you can actually determine the age of a person. And I'm sure that would be very useful in forensics and stuff. So you can look at, some specific islands in the genome that should be methylated at a certain age, and then maybe they lose that after a certain age. And you can actually distinctly tell quite a lot about the age of an individual with methylation pattern. So that's called an epigenetic clock. So if you guys are interested in reading about that, please go ahead and do that. It's pretty cool. Yeah. So now, if no one has any other questions, I'll move on to the next bunch of topics and then a couple more questions and stuff on that, right? So does anyone have anything they're confused about or anything they want to discuss? No? Okay. So all good then. I'll move on. So, uh, you guys read about the Hayflick limit, right? So this was discovered by a professor called Hayflick, and it actually disproved some concepts in the field at the time, which was that cells are actually immortal. That cells, if they're taken out of the body and cultured, they can just be cultured forever. And that was actually incorrect. And the person, there was a scientist who had, claimed that, but it turned out that, due to some experimental, you know, weird oddities that he had shown in his experiments, it was not a very accurate one, and that's why it had initially been shown that it's immortal. But actually, it is not the case. For normal somatic differentiated human cell population, basically, these are somatic means they're not germ cells, and differentiated means that they are not in the stem cell sort of pool. They've become a specific become a cell of a specific tissue type, right? And they have a certain function. So the human cell population will divide for a certain times until cell division permanently stops. So what Hayflick demonstrated was in normal human fetal cell population, and they'll divide between 40 and 60 times in cell culture before entering a senescence phase, meaning that they will stop dividing completely. And he interpreted his discovery to be aging at the cellular level, and he was kind of right, because we see a lot of these things in cells that are essentially becoming senescent over time in people who are aging, like as molecular or cellular characteristics. So this is one readout of that, which is the telomere shortening, like I mentioned. So you have, this is kind of the end of one of the chromosomes, and you see this telomere. And with progressive divisions, where the cell is doubling each time and doubling and doubling, you have this telomere becoming shorter and shorter until eventually it is totally gone, leaving this end very exposed and highly likely that it can undergo some fusion event or some DNA damage over here. And so at this point, the cell will shut down its division because it's no longer productive, and you will have senescent cells. So that's what Hayflick demonstrated, and it was a real like milestone when it came to understanding cells and understanding these various aging processes. So now I've been mentioning about, primary cells and how they have a certain limit to how much they can divide. But you guys, I think most of you are like bachelor's levels, right? So you guys must have, heard about some cell culture techniques where you have cell lines, and so you might have heard about, immortalization of cell lines, which means that they can just divide forever. Some treatment is given, some change has been made, so these cells actually can divide forever, right? And you've heard about cell lines, HEK cells, HeLa cells, blah blah blah. So how come they're dividing forever, right? Don't they have a Hayflick limit? So I thought I would just clear that up for you guys. So what is immortality, what is a transformed cell, and what is a primary cell? So you have normal primary cells. Okay, these are cells that have a finite number of cell divisions, and this is because of your Hayflick limit. So once their telomeres become very critically short, cells will become senescent, or they can become apoptotic depending on, whatever the internal sort of cell state is. And some examples of these type of cells that you can use within in culture are human dermal fibroblasts, mouse embryonic fibroblasts, or basically any primary cell that you can sort of culture for once it's taken out of the body, or ex vivo, you can culture it in your cell culture facility. That will have a finite number of times it can be passaged. Okay. Then now what are immortalized cells? So these cells can bypass the Hayflick limit, and they can actually divide indefinitely because there is this telomerase activation, or there can be other mechanisms that prevent the telomere from shortening. So one common way is it can occur naturally, or it can be induced, and this is something people do when they're making a cell line. They will express this HTERT or SV40 large T antigen. So basically what these do is that they have this excessive telomerase activity upregulation. And for instance, this HTERT, this is, I think, in the form of a virus, and you can infect your cells with it. This will integrate into the genome of the cell, and it will have this high telomerase activity, which will keep the telomere long forever. So some examples of cell lines that have been made immortal using these different techniques. So HEK293. Oh, I meant to write HEK293T here. HC40 large T antigen was used for HEK293T. So these are human embryonic kidney cells, and they are a very, very commonly used cell line for all sorts of different studies. Particularly, one important function of this cell line is to produce virus. So you use your packaging virus, packaging vectors and virus, and you infect this, and then you produce a lot of transfect it into this HEK293T, and you produce virus, final package virus like lentiviruses or retroviruses, and you can then use that to then express different genes in other cells. So HEK293T is a very common cell culture, um, uh, like cell line that you'd find in your cell culture. I'm sure you guys, if you have done cell culture work, you would have definitely come across it. Then NIH3T3, these are mouse fibroblasts. These are also immortalized. They look kind of like long cells. Then HACATs, these are human keratinocytes. Um, these are also immortalized. But now I wanted to just clarify for you guys that useful lifespan in the lab is not infinite. So it's not like if I have, say, a vial of HEK293 T cells that were frozen at passage 3, and then I just use them forever for my whole PhD. That is not how it works. Actually, after a certain point, you cannot use them anymore. Like for instance, in our lab, HEK293T, we don't use beyond, say, passage 22 or 21. Because what happens is that you accumulate these mutations in the cell line, and you can accumulate damage also, and stress from cell culture can actually cause the cell lines to not be very functionally viable after a certain point. So while they will divide, and in fact their division becomes slower, they will keep dividing. Their usefulness in the lab is not so much. So actually, despite them bypassing Hayflick limit, you can only use certain cell lines up to a certain passage number. Do you guys know what passage numbers and all all that are, just let me know. Otherwise, I can explain that as well. Do you guys know? You can let me know in the chat also, because if I'm saying this, but if you don't know what it is, it's not going to mean anything. Everyone knows, right, what a passage number is. If anyone doesn't know, you can let me know. I'll explain it, okay? So yeah, so as I was saying, the useful lifespan. Oh, someone says no. Okay. So, all right. So basically, it's a important cell culture term. So a passage number is essentially when a cell becomes, I don't know if I included it. No, I didn't. Okay, so when a cell, suppose you're growing a cell in a flask, okay, a bunch of cells, they are dividing and all that, they're adherent. So, or they can also be suspension, but let's just say you have some adherent cells that are sticking to the bottom of your T25 or your flask, okay, and you have let them grow for like a day, and now all of them have grown and they are what we call confluent. That means you cannot grow anymore because all the cells are touching one another. There's contact inhibition and they will stop growing. Okay, they'll stop dividing. That is when this cell that those cells are said to be confluent, that they've grown as much as possible within that confinement and they cannot divide any further because of contact inhibition. So what we need to do to preserve their health and preserve their function is that we need to do what is called passaging or splitting. What we do is we use some reagents to take the cells out gently, and you can and then you can divide the cells. So basically, you can use half of the cells and keep them back in that flask. So now you have half the cells there, and take the other half and you can throw it away or put it in another flask. So that allows the cells to have more space. So if you want to say keep your cells for a couple of days because you're doing a couple of experiments, you may need to passage your cells in between so that you have healthy cells that are ideally in log phase of division. Okay. So that's what passaging is. So now when coming back to this, suppose you have normal primary cells, they will have a very limited number of passages that they can undergo. Immortalized cells, typically much higher passages can be reached, the number of passages. So as I was saying in HEK293T in our lab, we don't go beyond 21 or 22 passage number because after that they accumulate a lot of errors. That means that 21 or 22 times from the start of when this cell line was revived or created, we can divide it, we can split the confluent cells and seed or reseed half the population or whatever population we want. Okay, so that's what a passage number is, and that's how it's relevant to cell culture. Okay, then the final thing here is the transformed cells. So these are cells that actually undergo some additional genetic alteration. So you have immortalized cells. So now these will be able to divide indefinitely. And then if there are further genetic changes after that, that's what makes them tumorigenic. So they're capable of actually forming tumors in vivo, meaning that if you have these cells in a dish and you take a whole bunch of them and you inject it into a mouse, they will go and form tumors or metastatic lesions. Okay. So they often show a loss of contact inhibition. You will not see this loss in immortalized cells, but you will see it in transform cells. So what would that mean? That once they have become confluent or you have the full capacity of your flask or dish that you're growing the cells in and they've all grown, they'll start growing on top of each other. They'll form foci, they'll form spheroids, depending on what kind of cell line you have. They might show anchorage independent growth. That means their anoic is resistant. They don't need to adhere to something. They can just float and grow. And they have high proliferation rates. And so this transformation can be because of some oncogenic activation. So you can have P53 mutations, you can have RAS mutations or RAS gain of function or MYC gain of function mutations. So that's an oncogene activation, or you can have TSG or tumor suppressor gene loss. So P53 mutants or RB mutants. And some examples of these are these cancer cell lines. So of course you guys have heard about HeLa cells, right? That come from Henrietta Lacks. So these are cervical cancer cell lines from a human female. MDMB231, this is a breast cancer cell line. It's an invasive triple negative breast cancer line. And E549, which is a lung cancer or lung carcinoma cell line. So these are all one step beyond immortalized. So they also have some other genetic changes that transform them and make them cancerous. Okay. So these are the three different types of cells and cell lines that you might come across when you start doing some cell culture work. Okay. So I hope that is now quite clear to all of you. It's very useful to know if you want to do lab work and TC work and so on. So after this, you guys, of course, listen to those very excellent lectures about apoptosis, and I'm sure it's very clear about the two different pathways. I thought I'll just recap in case it's been some time since you've seen the slides. Um, but as you know that apoptosis, it is a programmed cell death pathway, and it's an activation of a pre-existing genetic program. So it's already something that the cell is capable of, and in the time of need, it can activate this genetic program, and that results in its own death. It's a very evolutionarily conserved phenomenon, and it can be phenotypically seen as the cell shrinking. You can have nuclear condensation, right? You can have membrane blebs or blebbing, and then you can have DNA fragmentation and the release of these apoptotic bodies. And it is an active process, meaning that it needs ATP. So apoptosis is very important for homeostasis. That means normally in the body also, it's not a bad thing. Apoptosis is constantly taking place all the way from development, where it is involved with sculpting and making sure that all the right parts are being formed, and then like tissue in excess regions where it's not needed, it's being destroyed. And through time, apoptosis is very, very important. Okay. And dysregulation of apoptosis can, of course, result in a host of different diseases, one major one, of course, being cancer. Right. So you can have, too much apoptosis, too little apoptosis. These can lead to their respective diseases. Uh, so now you have two major pathways in apoptosis. One is your extrinsic pathway. So that you can see over here. So the extrinsic pathway of apoptosis is typically initiated with the help of these death receptors at the cell surface. They will come across this specific cognate death ligand. So what cognate means is that it is a molecule that is specifically binding to this. So receptor ligand interaction, where this receptor will only bind this ligand and vice versa, and they're very specific to one another. That's called a cognate interaction. Okay. So in the extrinsic pathway, you have these death receptors which bind cognate death ligands. This induces a conformational change in them that transmits through the cell membrane. Okay. So then you have some signaling that eventually leads to caspase activation and apoptosis. So what is really happening here? And this is a very fast pathway. I think it was mentioned, right, that this is a quicker way in which the cell can initiate apoptosis. Within a couple of hours, you're all done. So there are three major cell death receptor ligand pairs that have been described, and they are all members of this superfamily, tumor necrosis factor receptor, or TNFRSF superfamily. And so the first one is this Faz and Faz ligand. So Faz R, Faz L interaction. Then you can have these death receptors DR4 and DR5. And you can have also TNF related apoptosis inducing ligand or trail. So these are death receptors, and then TNF alpha and its receptor TNFR1. So these are the different receptor ligand pairs that are involved in the extrinsic pathway. So these are the ligands in the extracellular space. This is where the receptor is with its transmembrane domain, and it will interact here, and that will cause a conformational change. This is the death domain that is there in this death receptor. This is a conserved protein domain that's seen in them. And then this will activate the procaspase 8, which is the caspase involved in the extrinsic pathway. And that will essentially, the procaspase 8 is the inactive form, so that activates to the caspase 8 form, which is able to actually then go ahead and talk to these BH3 only molecules. So the BH3 only molecules are like, I think BIM, BIC, BAD, all of those, BID, and so on. They are the BH3 only molecules, and they're like the mediators of the apoptosis pathway. Okay, and then they will speak to the pro-apoptotic proteins like PAX and BAC. And that can lead to the yeah, and then that can then talk to the mitochondria, and that can go into the intrinsic pathway. That is later on. What can first happen is that this activated caspase 8 that can talk to effector caspases like caspase 3 and 7. They will get activated from their proforms, and they can then cause apoptosis downstream. And this disc, this is called the death inducing signaling complex. That is the sort of hallmark of the extrinsic pathway. And yeah, I think that's all for the extrinsic pathway. And of course, you have these inhibitors of apoptotic proteins that are going to inhibit apoptosis. You can have flip inhibiting this interaction and so on. So you have a lot of regulation on this as well. Then you have the intrinsic pathway. This involves heavily the mitochondria. Okay, so the mitochondrial pathway of apoptosis centers around one very important event, which is this mitochondrial outer membrane permeabilization, MOMP. And this is considered the point of no return. So if this has happened, then apoptosis will be induced one way or another. Okay, so typically, MOMP is normally prevented by anti-apoptotic members of the BCL2 family. So you guys read about the BCL2 family, right? And that there are anti-apoptotic members, pro-apoptotic members, and the BH3 only members that are sort of mediators between these various signals. So anti-apoptotic members have BCL2, BCLXL, BCLW, and so on and so forth. That can that and these proteins all have 5 domains, 4 BH domains, and a transmembrane domain. Pro-apoptotic, they have BAX and BAC and BOC also that are pro-apoptotic. They have one less BH domain, and they are important for inducing this mitochondrial outer membrane permeabilization. Okay, so what happens first? So you have a stimulus such as say cytokine deprivation, you can have DNA damage, irradiation, any kind of external stimulus that is interpreted as a chemical signal within the cell from its membrane. This will first activate some BH3 only molecules, and that leads to eventually the activation of BAX and BAC that can then talk to the mitochondria. So what happens here is that they go and they mediate this MOMP by forming some little openings in the outer mitochondrial membrane. So then what will happen? You will have the release of this mitochondria only protein, which is the cytochrome C, into the cytosol. Normally it's not there, but if it were to come out of the mitochondria, what happens is it binds to this protein in the cytosol, which is this apoptotic protease activating factor 1 or EPAF1 at some domain of it. Okay, so then cytochrome C will bind EPAF1, and this will then recruit caspase 9. So it will convert procas9 into caspase 9, and this complex is called the apoptosome. Okay, so once cytochrome C binds EPAF1, it promotes oligomerization and initiates apoptosome formation. This can then lead to the same target here, which is effector caspase 3 and 7 activation, leading to apoptosis. Okay. So I think that was actually a longer recap than I had expected. Yeah. Cool. So that's the extrinsic and intrinsic pathway, and I hope that revised it for you in your minds. So another interesting aspect of apoptosis is how it is important during homeostasis, and it can actually lead to morphogenesis in different organisms. So naturally, it occurs in organisms during various phases of their life, such as morphogenesis and embryogenesis. So one example you can see here is that it actually helps separate out the digits in your hands and in your feet. So we are not webbed, right? We all start off a little bit fish-like when we are developing as embryos, and we look a little fishy. But then after that, we start lacking these, we start getting rid of these webs in between the digits of fingers and feet. and they become individualized. So how is this happening? There are cells here, so they are essentially they get rid of with the help of apoptosis, and developmental biologists study these processes to figure out how is it that the body knows exactly which ones to get rid of. And this has been seen in people where there are some genetic mutations that people actually can be born with some skin remaining over here because there was some defect that led to a problem with respect to the morphogenesis, and they didn't have proper digit individualization. Another example is apoptosis when a tadpole is becoming a frog. So this tail they need to get rid of. So how does it happen? When it's becoming a frog, it is apoptosis. Another example is during Drosophila embryogenesis. There's actually some process called dorsal closure, where there is this gap that they need to close in the dorsal region of their body, and this is they they close this region by having some apoptotic cells in the middle, like that. Yeah. So if you guys are interested in reading about this, then I'm sure there are many interesting resources. This paper is nice too. You guys can check it out. But this is just a sort of um, homeostatic, developmentally important role of apoptosis. So I thought this is something that wasn't mentioned in the coursework, but it's useful for you to know. And if you want to read more about it, of course, it would be of interest to you, which is what happens after apoptosis, right? You have a cell that has died. It is contained and there's these apoptotic bodies, but are they just floating around in the body? Because eventually they might interact with something, or, they'll just build up, right? So someone has to get rid of them. So what's happening? So there is this process which clears these cellular corpses, even in homeostasis, in disease, and it is a phagocytic process, means that a cell is taking it up, eating it up, okay, of these dead and dying cells and apoptotic bodies, and this is called efferocytosis. Okay, this is a very important function. It's performed by immune cells like macrophages, and also to a lesser extent by other professional phagocytes such as monocytes and dendritic cells, and these non-professional phagocytes such as epithelial cells. So non-professional essentially just means something that doesn't do it as a part of its typical function. Um, yeah. So efferocytosis is important for clearing up this dead cell and all that. And how does it know that a cell is dying, or how does it know that a cell is already dead, right? So there are some signals that will tell a phagocytic cell that, hey, I'm dead, so please take me up and, recycle. And there are cells, but then, how would a cell know that, don't take up the cell? So there are actually some do not eat me signals as well. Okay. So some examples of eat me signals. There are actually some other signals called find me signals as well that I learned about recently, which involve some certain types of lipids on the surface that will signal to immune cells to come nearby. Then when the cell is dying, they'll have these eat me signals. So one very well-known example is this phosphatidylserine. So typically phosphatidylserine is found on the inner leaflet of the um, plasma membrane. Okay. So it's not externalized, it's not on the outer surface of the cell. But what happens when a cell undergoes um, like apoptosis is that you will have a higher amount of phosphatidylserine on the outer membrane, on the outer leaflet of the cell membrane. We know that the cell membrane is a bilayer, right? It's two layers of lipids. Um, So when this PS or phosphatidylserine comes on the outer membrane, it can actually interact with some receptors that are seen in these phagocytic cells. So you have PS interacting with receptors like TIM4, TIM1, so on and so forth. You don't need to know these, but basically there are receptors, multiple receptors that can bind to phosphatidylserine, and that will lead to some reorganization in the actin cytoskeleton and other signaling processes that can allow this phagocyte to then go and eat up this dead or dying cell. Okay. So those are eat me signals. Interestingly, we also have do not eat me signals, and that's something that is um, sort of taken advantage of by some, pathogens and cancer cells as well, because they don't want to be taken up by the immune system. One example is CD47, which binds to this SERP alpha receptor on phagocytes, and that actually inhibits. So these are inhibitory phosphatases, and they inhibit uh, the um, the phagocytic sort of signaling cascade and the phagocytic process by setting up these different suppression pathways via ships. So yeah, some examples are CD47, CD31, CD24, and so on. So I thought that might be interesting for you guys. I hope you found it useful. So you can read more about efferocytosis. There's some really nice papers about it. And it's something that people are studying more about now because it's a very important homeostatic process that would prevent excessive inflammation, right? It would go hand in hand with apoptosis. Right. So with that, I'll move on to the next set of questions. If anyone has any questions, do please feel free to stop me in between and ask whatever is on your mind. Okay, so let's start with the next question. What is the primary reason for the Hayflick limit in normal somatic cells? Anyone? All right. So, uh, okay. I hope you guys are still there. So the answer here is actually B, which is telomere shortening with each cell division. Ah, yeah, someone has said just now. Great. So, good. I hope the rest of you are there too. Please try to send in your answers and you can unmute and say also if you don't feel like typing, whatever. Okay. So, yeah, the answer here is B, telomere shortening with each division. Telomeres get shorter and eventually the cell will not divide anymore. Okay. So, that's one of the primary reasons for Hayflick limit in unimmortalized or non-transformed cells. Okay, which statement correctly distinguishes primary cells from immortalized cells?
So
Anyone?
A.
Yeah, good. So that's correct. It is A. Primary cells have a finite lifespan, while immortalized cells actually divide indefinitely. So I explained that, right? That they have high telomerase activity, typically immortalized cells, which allows the telomeres to be maintained. They don't shorten, so they can divide again and again. While that's not the case for primary cells. Okay. All right. I hope that is clear for everyone. Okay. Next question. Which statement best describes transformed cells? What do you guys think? Anyone? So transformed cells were like one step up after immortalized cells, right? So maybe that'll help. Yeah, that's good. So someone has answered correctly. It is A. Transformed cells have acquired genetic changes that enable uncontrolled growth and loss of contact inhibition. So essentially, they've become tumorigenic, right? And why the other options are not correct? So they're not the same as immortalized cells because while they can both divide indefinitely, transform cells have additional genetic alterations, right? C option, they don't necessarily only originate from primary cells. You can transform an immortalized cell. And D is you can have transform cells always maintain normal karyotypes and exhibit predictable growth patterns. Neither of these statements are true because if a cell has some mutation, say a p53 mutation, DNA damage and other genome protecting mechanisms are not at play properly. So of course you can have weird karyotypes. You can have aneuploidy, you can have all sorts of chromosomal abnormalities, and it's very common in cell cultures. If you were to sit down and karyotype, you know, all the cells in your culture of say some tumor cell like HeLa cell or something, you would definitely see some abnormal karyotypes. And they don't exhibit predictable growth patterns because they don't have contact inhibition, and so they just grow like crazy. Okay. Oh, another thing useful, interesting point is that, or maybe I'll come to that later. Basically, when it comes to growing, not only is this regulated by uh, contact inhibition, but also lack of nutrients. So even with low nutrients, cancer cells are still able to proliferate for quite a bit. And there are some reasons for that, which I will come to later, um, if I remember. So, okay, next question. Which of the following is a key reason for passaging mammalian cell cultures? So, this I explained just a little while ago. So, let me know if you guys have understood this, okay? Anyone? So, okay, maybe I'll just tell you the answer here. So the correct answer here is to maintain cell viability and prevent overconfluence, which can alter cell behavior. So as I mentioned, if your cells have grown to a level where they cannot divide anymore because there's no space, or they've used up all the nutrients in the media that was provided to the cells in your culture, in your cell culture, right? Then you want to make sure that you don't have such overcrowding or overconfluence. So you can take some of the cells out and reduce the number of cells in that dish. So that will preserve the cell viability and it will allow them to keep dividing and allow them to stay healthy. Because if your cells are overconfluent, they will not divide. This can alter the cell behavior and that can have an impact on your experiment as well, right? If your cells are suddenly not dividing or they are competing for nutrients, that can not only cause some sort of competitive mutation and sort of some crazy thing to start there, they can affect your experiment. So you want to make sure you're passaging your mammalian cell cultures correctly when you're doing this work. Okay. All right. Next question. Which of the following is not a characteristic feature of apoptosis? Anyone know? Yep. Good. I'm getting some answers. Does anyone else want to try? So you're right. It is So D is not, unfortunately, it's not a characteristic feature of apoptosis. So you can have chromatin condensation, and you can have DNA so you will have chromatin condensation, you will have DNA fragmentation, and you will have membrane blebbing. These are all normal features of apoptosis. But what we don't have is cell swelling and membrane rupture, because you have to remember that apoptosis is quite a controlled process and it is a programmed process. So this is what is meant to happen, and you're also making sure that the whole process is not immunogenic, in the sense proteins that are inside the cell that typically don't go outside, if they were to go outside, they'll cause inflammation. So in apoptosis, it's much more contained like that. It is not necrosis, which is where the cell swelling and membrane rupture can happen, and that can cause inflammation. Okay, so the correct answer here is B option, cell swelling and membrane rupture. Okay. Okay, the next question. How does necrosis differ from apoptosis? So I think I gave it away just now. So I hope you guys have understood, right? The difference between apoptosis and necrosis. That will help you answer this question. Let me know. Yeah, you're right. It is option C. Necrosis leads to cell swelling and lysis, which can cause inflammation. So it's an inflammatory cell death. Okay. So your so 14th, the answer is C option. Okay. All right. Next question. Which organelle is central to the intrinsic apoptosis pathway? Yep. Anyone else wants to try? So the extrinsic apoptosis pathway depends on your death receptor and death ligand interactions. So your FAS and TRAIL and TNF and all these receptor ligand pairings lead to extrinsic pathway, caspase 8 activation, and then apoptosis downstream. Intrinsic apoptosis pathway involves the mitochondria, right? Where you have pro-apoptotic proteins of the BCL2 family, like BAC and BACs, that go and make pores in the mitochondria, allow cytochrome C to leak out, which binds to APAF1, forms the apoptosome, recruits caspase 9, which then leads to eventually the uh, process of apoptosis. Okay, so this intrinsic pathway, the main organelle which is very important for it is mitochondria. Okay, so the option B is correct here. All right, so which protein family regulates apoptosis by controlling mitochondrial membrane permeability? So I think I just mentioned that. Yeah, great. I think you guys know this well now. So it's the BCL2 family, right? So great. I think that's clear. I don't know if there are more questions. Okay. 16 के बाद 13 कैसे आ गया बे? Okay, I have done some numbering issue over here. Don't worry about that. Okay, so the next question is, what is the role of caspases in apoptosis? Anyone? Yeah, so the right answer here is that they degrade cellular components and drive apoptosis. Okay, we know that the caspase family of proteins is very important. You have your initiator caspases and your effector caspases, right? So these subsets of caspases are involved in apoptosis. So you have your both your extrinsic and your intrinsic pathway ultimately recruit an initiator caspase, convert it from the pro form to its active form, which then goes and activates the effector caspases, which then lead to a whole host of different protein cleaving and so on and so forth that degrades cell components and drives this process. Okay, so the option that's correct for this question is B. Okay, so which caspase is a key executioner in apoptosis? Yep, I've got one answer. Anyone else wants to try? So you're right, absolutely. It is B option, caspase 3. And then caspase 7 also is uh, an executioner caspase, and 8 and 9 are actually initiator caspases. Okay, so good. I think that has been recapped well enough till now. What is the function of these apoptotic bodies? Can someone tell me? Yeah, I've got one right answer, two right answers. Great. So you're absolutely right. The answer here is C, that they package cellular fragments for phagocytosis, or the process that I mentioned, the specialized type of phagocytosis called efferocytosis. Okay. Okay. Great. Okay, next question. Which apoptotic pathway is triggered by external signals like Faz-L, Faz-R binding? This is an easy one that I made a little too easy by saying external signals. Yeah, so you're right. This is, yeah, evident that it's the extrinsic pathway. Okay, so you can have these death domain containing receptors bind to their cognate ligands, and that those are external signals that can trigger the extrinsic pathway of apoptosis. Okay, glad that that's clear. I think, oh my god, I have a lot of questions. Okay. So, which gene in C. elegans is an essential executioner of apoptosis and has a mammalian homologue in caspases? So, this was something that was sort of explained in the apoptosis part, where, uh, yeah, where this was being discussed, that initially this was something found in C. elegans, and then they found, because it's quite a conserved process, they found the respective orthologs in humans. So, what is that gene in a worm? Do you guys remember from your coursework? It's good to know. Anyone? Okay, I've got one answer. You're right. Yep, that is, it's said 9. So good. Um, this is just sort of like a knowledge question, honestly. So great. Okay, next question. Which of the following is an anti-apoptotic protein in the BCL2 family? Okay. Does anyone else want to try answering? Okay, so the right answer here is C option. It's BCL2. Okay, so BCL2 family it's called because that was the first one found, but actually BCL2 itself is an anti-apoptotic protein. Okay, along with BCLXL, BCLW, and so on. Pro-apoptotic proteins are this BAX, BAD, and so on. And BID is one of those BH3 only proteins. These are all part of the BCL2 family, but they can be either anti-apoptotic, pro-apoptotic, or mediators. Okay, so BID is a BH3 only protein that acts as a mediator. BAC and BAX, they are both pro-apoptotic, and anti-apoptotic in this case is BCL2. Okay, so it's option C. All right. What distinguishes anti-apoptotic BCL2 family proteins from the pro-apoptotic ones? So I mentioned this earlier and it's in your coursework as well. Remember those sort of gene uh, diagrams that the professor had shown? That might help you remember. Yep, so I've already got one answer. Does anyone else want to try? Yes, so great. So the answer is A, presence of an additional BH4 domain. So anti-apoptotic proteins of the BCL2 family have a BH4 domain along with the BH3, 1 and 2 and the transmembrane domain. And pro-apoptotic ones, they only have BH3, 1 and 2 and the transmembrane domain. Okay. So that is the correct answer, E option. All right, next. What is the function of BAX and BAC in apoptosis? So I just told you what BAX and BAC are, what kind of function they have as a part of the BCL2 family. So what do you guys remember about what it does in apoptosis? Hmm. So that's right. The option is C. It forms channels in the mitochondria. This allows cytochrome C leakage from the mitochondria, leading to the um, intrinsic pathway of apoptosis. Okay, so BAX and BAC are both pro-apoptotic BCL2 family members. Okay, so I hope this is now clear and you've sort of committed this idea to memory because it's very useful information for when you're trying to understand the process of apoptosis. Okay. I think I have three more questions and then just a little topic and then we're done. Okay, so what role does P53 play in apoptosis? This must be uh, relatively clear for you. So actually the answer here is D. Okay, so it can do all of these things. It can activate BAX directly. It can translocate to the mitochondria and it can act as a transcription factor for BH3 protein. So I think it was mentioned in the coursework that active P53 can be a part of apoptosis in two different ways. There's a transcription dependent way where it acts as a transcription factor. So P53 is a transcription factor. It acts as that and it can lead to the transcription of BH3 genes like NOXA, PUMA, etc. And of course, that leads to BAX gene transcription. which will lead to apoptosis. It can also act in a transcription independent way. It can go to the mitochondria, directly activate BACs, and help with cytochrome C release. So, um, the answer here is D, all of the above, that it can do both of these, play both of these functions. Okay. All right, next question. What is the role of IAPs, or inhibitor of apoptosis proteins? This is a fairly evident one. From the name only, you can kind of figure out what might be going on. Yeah, great. So that's correct. It is to inhibit caspases. So they do that at various stages. Um, yeah. All right, and the last question before our final topic is, why do neurons, was mentioned right in the coursework, retain some apoptotic machinery even though they restrict apoptosis? What is the point of doing that, right? Do you guys remember the reason? It's a delicate balance of apoptotic machinery that's needed. Hmm. So the answer is actually A. Okay. So it is not D because neurons don't actually divide. So there's nothing about regulating cell division really here. And B and C are, um, not right because, I mean, it's just not what happens here. The actual answer is A. It's to allow synaptic and axonal pruning. So as it was mentioned that neurons are connected to each other, right, at synapses, and they have axons and they have dendrites and so on. So to be able to remodel these interactions, they actually require a lower level of apoptotic machinery to be present there so they can sort of change and make little changes to the synapses. Okay. So that would require the apoptotic machinery, but neurons should not undergo apoptosis, even though technically they don't divide. One would think that okay, eventually they will undergo, but they restrict that, but use a few components from the cycle of apoptosis, from the cascade of apoptosis, for apoptotic, for axonal pruning and synapse remodeling and restructuring. Okay, so actually I have a bunch of slides left. I might go over a little, so I hope you guys stick around if you want to. Uh, because I thought I'd go into the hallmarks of cancer, but all of this took a little bit longer. So, as you know from this very famous paper from Hanahan and Weinberg, where they discuss the hallmarks of cancer. Now, this has been updated a little bit with a few more things recently, but basically, what are some of the hallmarks of cancer? Right, so one is that you'll have sustained proliferative signaling. You have mutations that allow the cells to keep dividing. They evade growth suppressors, they avoid the immune system, they avoid immune destruction, they hide from the immune system. They enable replicative immortality, so they may have some mutations that allow the telomeres to be unnecessarily long, even though the cell has divided many times. You can have tumor promoting inflammation. So this is inflammation that actually, at a low level, that actually helps the tumor to grow. It can cause vasculogenesis and so on, which is here. It can induce angiogenesis. You can have invasion and metastasis, where the tumor will move from a primary site to a secondary site and form metastatic lesions. You can have genome instability and mutations. This is very well known in tumor populations. They resist cell death, so they will have anti-apoptotic machineries that prevent them from dying, even though they're accumulating double-stranded DNA breaks and mutations and so on and so forth. Uh, you can have cellular uh, energetics that are changing. So you read that the tumor cells actually become more glycolysis dependent and downregulate OXPHOS in them. And also they manage to survive in highly hypoxic environments, which is something I meant to tell you guys earlier. and they will yes, and yeah, so those are all of these points, and this all contributes to how well a tumor can survive, how it can grow, how it can spread. Okay, so these are some of the hallmarks of cancer. I thought I'd talk about contact inhibition because I mentioned this quite a lot while we were looking at previous slides. So contact inhibition is what enables non-cancerous cells to cease proliferation and growth when they contact each other or when the cells are touching. So this is seen in most epithelial cells. It's associated with cell division halting, and it will initiate differentiation eventually. So what can happen is if you have a loss of contact inhibition, it can lead to uncontrolled cell growth, which is very characteristic of these solid tumors, and it can increase the ability also of cells to invade host tissues, in which is the process of metastasis, okay, where they escape from the primary site, move through the body without being detected by the immune system, and then set up shop in some other tissue or in some other secondary site in the body. And there can be dysregulation of these mechanisms where there is transmission of mechanical information and also this proteins in the adherence junction. So what is mechanical information here? If you have cells, spreading on a surface, stretching, pushing against each other, the stiffness of the surroundings, these all have effects on what is known as mechanotransduction, which is the cells can actually feel the stresses on them. They can feel tension, pulling, pushing forces. These are actually sensed by cells. Okay. So tension across these cell junctions and at focal adhesion points can actually affect the signaling within the cells. This GAPTAs, these are some transcription factors that are important for mechanotransduction. They're part of the hippo pathway. And this pathway has been found to be dysregulated in some cancers where you have a loss of contact inhibition. And another thing about another process in which this loss is there is by the loss of E-cadherin. E-cadherin is an important protein found in this adherence junctions between cells, right? And normally you will have these junctions with E-cadherin, and then when you have these cell-cell adhesion points, that actually inhibits from it going from a monolayer to just piling up cells one over another, right? So it'll just stop here. If you lose E-cadherin or you have mutated E-cadherin, actually it will not sense this. And I think there is a crosstalk between, the actual molecular signaling as well as the mechanical signaling. And that leads to this crazy adhesion and these piles of cells, okay, and they grow on top of each other. So there are various different mechanisms as to, why contact inhibition loss, how it really works. And these are some of those, okay. So the tumor, as we know, has a microenvironment, and the tumor can be designated in crude terms as cold or hot based on what kind of cells are there. Okay. So in this case, this is a cold tumor or hot tumor. So it's not actually talking about the temperature of the tumor. It's talking about the kind of cells that manage to infiltrate or get into this tumor. Typically in a cold tumor, you will see cells that they might be immune cells only, but they are immune suppressing in the sense they will suppress the anti-tumor activity of say T cells, right? So you might have these cells that go into the tumor cell and into the tumor microenvironment, and because of some secreted factors, they actually start supporting the tumor. And so you have less infiltration and less amount of these anti-tumor response elements like T cells and like effector T cells called cytotoxic T lymphocytes. Okay. So these tumors are designated as cold tumors, and typically the prognosis for patients who have cold tumors where you have less infiltration is not as good. So the target would be in this case would be if you want to have some design some therapy, immunotherapy or so on, you'd want to make sure these cold tumors become hot tumors, in the sense you have a lot of T cells and other anti-tumor cells that are able to infiltrate into this tumor and cause a lot of tumor killing. So you want to convert a cold tumor to a hot tumor that attracts a lot of immune cells, causes a lot of cell death, and then there's this feedback that sort of set up here a loop of like killing cells, calling more, killing cancer cells, calling more immune cells, killing cancer cells, calling more immune cells until you get rid of that tumor completely and have a memory of that tumor stored as well in case the tumor tries to grow again in the body. So therapies should make sure that they are trying to combat these cold tumors, and make them into hot tumors such that so the body can get rid of them better. Okay. So this is basically a depiction of that you can have cold tumors where you are inducing an immune response, but because of the lack of tumor antigens, a lack of priming, of dendritic cells via dendritic cells at the lymph node, you don't have good T cell activation. That can lead to impaired trafficking, meaning that they don't reach the tumor that well. That can lead to impaired infiltration into the tumors. So you have a cold tumor, whereas the hot tumor is you're removing these breaks. So you have different techniques in which, and you can read more about these in detail. You can have checkpoint therapy, PD-1, PD-L1. You can have some loading of dendritic cells, which improves how well you can activate your T cells, activate your adaptive immunity, NK cells even, to go find the tumor and kill the cells without being stopped by these different processes and different tumor evasion tactics that the tumors will employ to hide from the immune system. Okay, so that is a hot and a cold tumor, and these are some immune evasion strategies. You can read about these in much more detail. There are a lot of lovely papers with respect to that. One example, tumor heterogeneity. So you know that tumors have a lot of different antigens. So you need to be able to tackle all of these different heterogeneous cells. So then tumors are smart. So sometimes what they'll do is they'll get rid of the antigens on their surface, making them less visible to the immune system. And so then it tries to hoodwink the immune system in that way. It can downregulate antigen presentation and hide from um, the immune system by reducing the number of major histocompatibility molecules that are important for antigen presentation. It can exhaust immune cells, it can regulate them, it can inhibit them using checkpoint activation. It's resistant to apoptosis, so it doesn't die properly. It can mask its antigen, it can switch its lineage, it can induce immunosuppression in the environment, it can cause cells to become pro-tumor. Uh, it can enhance regulatory T cell function, so that goes and inhibits other T cells. It can induce these cells. These are tumor-associated macrophages and myeloid-derived suppressor cells. The tumors essentially make them pro-tumor, so they start supporting the tumor function. Then the microenvironment has a lot of these immunosuppressive markers, the physical barriers. There's a dense tissue that secrete a lot of extracellular matrix is deposited. You have these weird blood vessels that are very different from normal blood vessels in the tumor. They're very tortuous, very leaky. And then you have these different immunosuppressive cells in the environment that can inhibit your immune cells that are actually trying to get rid of the tumor. And you can read about what exhaustion means with respect to T cells and CAR T cells. Okay, so this is just something I thought I'd go into. And the last slide I had, sorry, it's going over time, is an interesting case. So this is about the Tasmanian devil. So typically you guys have heard about cancers not being communicable, right? So if someone has a cancer, it will not necessarily transmit to the next person in the room, right? If they were to say contact each other in some way, not necessarily. There are sexually transmitted viruses that can lead to the higher chance of developing a disease like HPV and so on. But directly, a tumor or a cancer like that has been discovered in this Tasmanian devil. This is this little animal that's found in Tasmania over here, and they have this strange facial tumor. First one variety of it was DFT1 that was found around then, and then there's a DFT2 that was found more recently. And these are associated with the population decline of these animals. I was reading that it went from like 60,000 to like 16,000 kind of a number. Okay, so really there was a huge population decline because of these facial tumors that you can see here. And they are from Schwann cell cancers, and there's some genome rearrangements that actually have, for instance, in DFT1, you have lower MHC1, which means there's lower antigen presentation. So maybe that's how it's escaping from the immune system. And what's crazy is that this is actually a communicable cancer. So this spreads between animals, it's not hereditary. And you can read more about, sort of what is the way in which people have been trying to figure out a vaccine and figure out a treatment regime to try to conserve this population so that they don't all spread this cancer amongst themselves and die. So that was about the Tasmanian devil. I hope that was interesting for you guys. Um, right. Okay. So that was it. Please let me know if you have any other questions and anything you want to discuss. If everyone's fine, then that's it for today. Thank you all for joining, and I'll see you next week for the last discussion session of this course. Cool. All right.